The honeybadger, that science still cannot explain. Fear is one of the most ancient survival mechanisms in vertebrates. It shapes behavior, avoidance, learning, memory. Without fear, animals die, yet honeybadgers appear to experience fear differently. Behavioral studies show minimal avoidance responses to predators. They do not flee from lions. They do not retreat from venomous snakes. They do not abandon food sources when threatened. Instead, they escalate. Neuroimaging of preserved brain tissue reveals reduced amygdala activation patterns compared to other carnivores. The amygdala governs fear processing, threat evaluation, and risk aversion. Honeybadgers appear to process threat without emotional amplification. They detect danger, but do not generate fear responses proportional to risk. Instead, their prefrontal control circuits remain dominant. This allows decision-making without panic, without retreat bias, without stress-induced errors. This neurological profile resembles neither predators nor prey. It resembles something closer to deliberate cognition, but in a mammal with a brain the size of a walnut. This should not exist. High-level emotional regulation typically correlates with large cortical volume. Honeybadgers violate this correlation entirely. Extreme aggression, continuous movement, pain insensitivity, injury tolerance, all of these require energy. Enormous energy, yet honeybadgers operate at metabolic levels that should be unsustainable for animals of their size. They roam huge territories, engage in prolonged combat, dig through hard soil, tear into beehives, and survive on erratic food sources. Metabolic measurements reveal unusually efficient mitochondrial function. Their muscle fibers demonstrate elevated oxidative phosphorylation capacity, and their ATP recovery rates exceed those of similarly sized carnivores. But here is the mystery. High metabolic output usually accelerates aging. Oxidative stress damages cells. DNA accumulates mutations. Tissues degrade. Yet honeybadgers exhibit long lifespans relative to their body mass. Field studies suggest individuals can survive over 25 years in the wild, far beyond what metabolic scaling laws predict. This implies cellular repair mechanisms operating at unusually high efficiency, possibly involving enhanced telomere maintenance, superior autophagy pathways, and mitochondrial quality control. But no complete genomic model explains how these systems coexist without trade-offs. High energy output without accelerated decay should not be possible. Yet honeybadgers defy this constraint. Most animals survive through avoidance, stealth, speed, or social coordination. Honeybadgers survive through confrontation. They do not optimize for safety. They optimize for dominance. Even when outmatched, even when injured, even when escape is available, they choose engagement. This violates basic evolutionary risk modeling. Aggressive confrontation increases mortality probability. Yet honeybadgers persist. And not only persist, they thrive across deserts, forests, grasslands, and savannas. Behavioral ecologists attempting to model honeybadger decision-making using standard game theory frameworks failed. The animals consistently selected strategies with higher immediate risk but higher long-term resource acquisition. This suggests their survival strategy operates on a different payoff matrix, one where dominance and deterrence outweigh energy efficiency and risk avoidance. Over time, this forces competitors to yield, predators to disengage, and resources to become accessible. In effect, honeybadgers do not adapt to the ecosystem. They coerce the ecosystem to adapt to them. No other mid-sized carnivore exhibits this behavior pattern consistently across environments. Long before viral videos, before field cameras, before academic journals, indigenous communities across Africa told stories about the honeybadger, stories that modern scientists dismissed as exaggeration, until they began witnessing the same phenomena themselves. In some regions, the honeybadger is called the deathless one. In others, the eater of snakes, or the spirit that walks through fire. These stories describe animals that survive venom, ignore wounds, walk away from predators, and return again and again. In traditional folklore, the honeybadger was often portrayed not as an animal, but as a trickster, a disruptor, a creature that violates rules. The interesting thing is not that myths exaggerate animals. The interesting thing is that these myths align disturbingly well with empirical observation. Survival after venom, fearlessness toward predators, extreme resilience. These traits were documented long before science explained any of them, which raises an uncomfortable question. How often does folklore preserve biological truth before science catches up? And what else might we be ignoring because it sounds implausible? Evolution operates through trade-offs. Strength costs speed. Armor costs mobility. Aggression costs longevity. Resistance costs efficiency. Yet honey badgers violate these trade-offs repeatedly. They combine durability with agility, aggression with longevity, pain tolerance with survival, high metabolism with extended lifespan. These combinations should not persist. Natural selection should eliminate unstable strategies. Yet honey badgers remain remarkably conserved genetically across populations. Their traits are not localized adaptations. They are species-wide constants. This suggests that their biology occupies a rare evolutionary basin of stability, a configuration where normally incompatible traits reinforce rather than undermine each other. But no current evolutionary model predicts such a configuration emerging without intermediate collapse. No fossil record shows transitional forms with partial versions of these traits. There is no evidence of gradual ramping toward this phenotype. It appears suddenly, fully formed, and persists. This pattern resembles evolutionary punctuated equilibrium. But even that does not fully explain the absence of unstable intermediates. It is as if honey badgers jumped across a fitness valley without passing through the lethal zones in between, which should not be possible. When researchers examined honey badger neurotransmitter profiles, they found abnormal distributions of dopamine, serotonin, and norepinephrine, neurochemical signatures associated with motivation, reward, stress resilience, and fear suppression. Dopamine turnover rates were elevated, suggesting enhanced reward processing during high-risk behavior. Serotonin receptor density patterns differed from other carnivores, associated with impulse control without withdrawal. Norepinephrine levels suggested dampened stress reactivity under threat. Together, this creates a neurochemical environment optimized for sustained confrontation, minimal fear, minimal retreat, and continuous engagement. But here is the anomaly. These neurochemical profiles usually correlate with pathological states in humans: mania, psychopathy, impulsivity disorders. Yet in honey badgers, they produce functional survival strategies, not dysfunction, not instability, not self-destruction, but adaptive dominance. This suggests that honey badgers operate under a fundamentally different neurochemical balance regime, one that in other species would cause collapse, but here produces stability, which again violates comparative neurobiology expectations. If you enjoyed uncovering these amazing animal secrets, make sure to hit that like button and subscribe to stay updated on all our future videos. Don't forget to click the bell icon, so you'll never miss out on more fascinating stories from the animal kingdom. Thank you for watching, and we can't wait to explore more hidden secrets with you in the next video.